So our next speaker is Dr. Annegret Ulke-Lemme, a senior research associate from Justin MacDonald's lab, and Justin is her mentor. And Anna is going to be talking about how we can detect many proteins in one sample without a single antibody.
So I'd like to talk to you about an exciting new method I'm working with, which allows us to detect many proteins, up to 100, in a single sample in about an hour. And no, there's no need for any antibodies. So all of us who work with cardiovascular research to help cardiovascular patients, most of us are working with people or with some sort of model systems. And then we extract, in order to get data on our patients or model system, we are using some biological samples or maybe some biofluids. And all of those, sooner or later, end up in a test tube. And we're asking, what is in this test tube? We want to know about proteins, because proteins is what the activity of the cells are actually doing, or the change of proteins is the outreach of the activity. So what kind of protein is in there? How much did it change? The typical way of doing this is to use antibodies. So about a certain proportion of this audience has been doing Western blottings or ELISAs or the newer Luminex to quantify my protein and to detect it. In the case of Western blotting, we are limited to maybe 5 proteins per sample, if you're lucky. In the case of ELISA, it's very few. In the case of Luminex, that number goes maybe up to 20, but that's it. The thing is as well, antibodies, well, they're really only ever so good. These are an example of my very beautiful Western blots throughout the years. So sometimes we see several bands. We have no idea which one of those is my protein of interest. More often than not, we see no bands, or just some plain ugly results. Or most of the time, the antibodies for my protein of interest is just plainly not available. Just it doesn't appear to create an antibody. So now what are we doing now? Well, I have this sample. I really want to research cardiovascular research. I really want to know something about my proteins. So in comes a new method. Since about 2003, there was a flurry of activities in the world of mass spectrometry. There has been improvement in mass spectrometry instrumentation, methodology, and the software. And this led to the development of multiple reaction monitoring mass spectrometry, which was called MIMMS. MIMMS was termed Nature Method of the Year for 2012 for its potential to really revolutionize medicine. So we are applying MIMMS because it can measure so many proteins at the same time. So it can be multiplexed. I can see 100 proteins, or up to 100 proteins if I'm lucky, in a single sample, single experiment. There's no need for any antibodies. And importantly, the only information I need to detect this protein is its amino acid sequence. And ever since we got the genome sequence, I know the amino acid sequence of my protein of interest. Importantly as well, this is targeted, so I can pick which protein I would like to detect with this method, and I go ahead, design my method, and look for it. I don't need any other information about this protein, and I certainly do not need an antibody. I can use all kinds of biological samples I would like to use. So in my case, I'm using urine. People have been using blood samples, tissue culture samples, and so on. So in order to do MIMMS, one needs this beautiful machine called high-performance liquid chromatography triple quadruple mass spectrometer, or in short, LC-MSMS. So this is a machine I personally work with, which belongs to Justin MacDonald and Ed O'Brien. So the LC-MSMS works as a filter mechanism. It's like a series of sieves. So what I'm doing is when I have my protein of interest, whatever you happen to be working with, I define myself a prototypic peptide. So this peptide is derived from the protein and is unique to this protein. Because I know all of the protein sequences, I can pick a peptide which is unique to my protein of interest. So what I do now is I have my protein soup in my test tube. I digest all of these proteins into peptides, and I feed it all into my mass spec. The first selection step is the hydrophobicity of this peptide. So the HPLC number 1 selects all of my proteins by hydrophobicity. The hydrophobicity of the peptide is known because I know the peptide sequence. So that's my very first selection criteria. Importantly, this hydrophobicity is very reproducible from one run to the next. So the now selected peptides gets fed into the mass spec, For second selection criteria based on the mass to the charge of the peptide. Again, the peptide sequence is known for which I can calculate its mass, and I can calculate the charge based on the amino acids in that particular peptide. So the mass spec selects out only those peptides which meet my specific mass to charge ratio, which are pre-select. So that's my second selection criteria. After these two selection criteria, I'm already down to maybe between 20 to 100 peptides. That's a very small number considering I'm starting with a giant soup in the beginning. So now comes my third selection criteria. I take these peptides and I fragment them further into small parts. Conveniently, peptides fragment along their peptide bonds, so I get bits of amino acids. Again, the amino acid sequence is known. I know the size of these fragments, and I can pre-program the mass spec to detect these particularly mass to charge of the peptide, of the fragment in this case. So I have three selection criteria in a row I'm using to detect the proteotypic peptide derived from my protein. So 3 selection steps. If you think back about Western blotting, I have 2 selection steps. I select based on the size of the gel as well as an antibody. In the case of ELISA and Luminex, I have 2 selection criteria, typically 2 antibodies. In the case of MIMMS, I have 3 selection criteria. I have the hydrophobicity, the size of the peptide, and the size of the fragment of this peptide. The other important thing is the mass spec is so fast that I can detect very many of these prototypic peptides in a single second. So I set the entire mass spec to detect one prototypic peptide, then I cycle and detect the next one, and I cycle and detect the next one, up to very many at the same time, which is somewhat limited currently on the speed of the mass spectrometer. So I can multiplex it up to, has been reported, 100 proteins from 100 different peptides. Importantly as well, I can increase the sensitivity and the specificity by using more peptides per protein. Proteins are fairly large. They give me 5 or 10 or 20 peptides. I can try to find all of those, and now have very high specificity because I can say I want to find all of those peptides. Again, antibodies usually goes to 1 epitope, and that's it. So now where does quantification coming in? So now that I have the 3 selection steps, I can find my peptide, but I want to quantify it. So once I know my prototypic peptide, I synthesize the same peptide with a heavy labeled carbon. If you remember, the carbon-13 is 1 atomic mass unit heavier. So now the mass spec can recognize this heavy labeled peptide and distinguish it between my endogenous. So I use this heavy labeled peptide and spike it into my sample as an internal control at a known concentration. Now I run my method, and the mass spec can distinguish between those two, quantify both, and because I know the concentration of one, I get the concentration of the other. And the prototypic peptide stands directly in for the protein it's derived from, so I get an absolute concentration with this method. So something really exciting I am doing, applying MIMMS in cardiovascular research, is we are looking at people with kidney disease. When people have acute kidney injury, they either go on and recover, or they go on and develop chronic kidney disease. And we want to know why. So what we are doing is we have a set of about 20 disease-associated proteins based on our own research, and based on literature, we think these proteins are involved in acute kidney injury. However, out of these 20 disease-associated proteins, 2 have an antibody, and that's it, and no other antibodies inside. So now I can program an MIMMS for that and actually quantify exactly these proteins in my sample. Along goes a couple of control proteins because I can run as many peptides as I want. I'm going to throw a couple of controls in, and a couple of biomarkers which have been published to maybe playing a role as well. So the way these goes in as well. So now I have 30 proteins I'm detecting. So I'm collecting the urine from patients with AKI and detect these 20 proteins, quantify them, and then I can go link back which of those are involved in the development of chronic kidney disease or not. That's really important for biomarker discovery, also as a future drug target. So in summary, MIMMS is here to stay at the University of Calgary. Other than looking for biomarkers in AKI, there's a lot of other things you can do, such as looking for phosphorylation, looking for single nucleotide polymorphisms, or really whatever else you can think of. MIMMS is going to be very important for precision medicine. We're going to hear of that later. And there's still a flurry of activities further improving the field of MMS, which will make this field even more important in the future. Thank you.
Thank you for making mass spectroscopy understandable. That's no easy task. So any questions for Anna about this technology?
And I was just wondering, do you think this is a technology that's going to be amenable to, let's say, hospital diagnostics labs and places where this sort of detection and quantification could really help and replace some of the technology that currently exists?
So Andy, I think that's certainly the future. Using this very similar method to detect small molecule is already very, very common in clinical diagnostics. So the method is extremely quick. Between getting a sample and having the data, it's not even maybe a day. And I think the future really is to do that in a clinical setting. It's also fairly cheap. And it's certainly going to replace ELISAs or like really compete with those. Yeah.
And one last question here.
How much does the machine cost and how much does it cost to do a sample?
A sample is between $10 and $20 per sample. It depends a little bit on the preparation. The machine, you'll have to ask my boss. It's not cheap. It's about the value of my house, I would think, plus minus. Plus another. Okay. The value of his house?
House in Vancouver. Thank you very much, Anna. That was fantastic.